Alright guys, today we're going to cover everything that you need to know step by step about cellular respiration. And this is going to be covering pretty much all the material you would find on a test for AP Bio or early college bio. So cells need to store energy. A lot of the living processes that occur in a cell require energy. Cells can't just release all their energy at once. They need to be able to store it and control that release of energy for specific purposes. And they store that energy in organic molecules. Organic means having carbon in it and being used for living purposes. Cells need to harvest this energy from the organic molecules because they stored it away, and now when it comes time to need to use it, they need to take it out. And that is what cellular respiration is. So it's cells harvesting the energy from organic molecules. Now, as far as AP and college bio go, the organic molecule that gets used for cellular respiration is going to be glucose. And from now on, I'm going to use its formula right here. So we get used to associating that formula with the name. And glucose, what's going to happen overall to glucose is that it gets chemical energy gleaned off of it. And that chemical energy comes in two forms, ATP and NADH. And NADH actually gets used later to create even more ATP. We'll see what these are and how that works a little bit later. And then the glucose is just going to be released as CO2. So these forms of chemical energy gleaned off it, let's take a look. And let's start with ATP. ATP is a molecule called adenosine triphosphate, and it's a molecule used as chemical energy in cells. Now, this is a really oversimplified structure of ATP, but it works for our purposes because we can see triphosphate, the three phosphates right here. Please note that these phosphates are not just pure phosphorus; they are phosphorus with a bunch of oxygens around them. These circles with P's are just ways to represent the phosphate groups. And the way it works as chemical energy in the cell is that this molecule can be taken anywhere. Let's say a protein needs a little bit of energy to do a specific process, and then the ATP just goes up to it, and then has its last phosphate broken off, and then energy gets released. And then the protein can use that energy. And then later on, this ATP can go up to an energy source of some kind and have its phosphate put back onto it. So it's recharged. So then it can be taken somewhere else to release more energy if needed. And that's how ATP works as a source of chemical energy in a cell. Now, we're going to look at our second form of chemical energy, NADH. NADH as a molecule is just an electron holder. And before we actually look at NADH, let's look at an important concept in bio. If I have a molecule with less pull over an electron, and right next to it a molecule with greater pull over electrons, outermost electron that this molecule has, it's going to want to go over to the molecule with greater pull over the electrons. And the electron wanting to do something, it's like a ball at the top of a hill. If the electron has yet to do something and this greater pull is inspiring it to do it, that means that there's potential energy in this system. By system, I mean just like imagine there was a little box around this molecule, and this electron. If this electron hasn't yet traveled, it has potential energy. And this potential energy is going to get released when this electron goes over here because now the whole system is more stable, and that releases a bit of energy. How is NADH used as a form of chemical energy? Well, we can see how the electrons can be used to release a bit of energy. So NADH works as chemical energy by being an electron holder. Later on, we're going to see it take a bunch of electrons over to a system that works like this to release a bit of energy. This NADH, this energy that's released, by use of NADH is actually going to be used to make even more ATP, which is something that we'll see in a bit. So we got to answer, how does NADH take electrons from the glucose? And that is using an enzyme called dehydrogenase. Dehydrogenase is aptly named because it takes two hydrogen atoms from the glucose. And by the way, guys, an enzyme is a protein that helps a chemical reaction, such as this one, happen. Dehydrogenase is aptly named because it takes the two hydrogen atoms from the glucose and gives it to NAD+. And if you guys need a little refresher on chemistry, a hydrogen atom is just a proton and an electron surrounding it. So by taking away two hydrogen atoms, it is also taking away two electrons. If you look closely, the NAD plus is actually leaving an extra proton just to the side, because if I take one electron, that turns the NAD plus into just NAD, because plus means that it's actually missing some electrons. If I take one electron, that turns it into the NAD, and if I take one electron and one proton, that's how we get NADH. But that means that we have actually one proton left. And I want you to remember that this one extra proton, it's gonna be involved in cellular respiration later. So don't forget about this one extra proton. So every time you see dehydrogenase at work and how it gives electrons from glucose to NAD plus, I want you to remember that there's actually one little proton that's going to be released because of that. This overall reaction where glucose is getting electrons taken away from it is called a redox reaction. And there are two terms that I want you to know. Oxidation is when something gets electrons taken from it. An example would be our glucose is getting oxidized. Reduction is when something gets electrons given to it. So our NAD plus was reduced. And a fantastic acronym I learned in school was oil rig. So oxidation is loss of electrons and reduction is gain of electrons. And that just helps you memorize those terms. So quick recap of our little bird's eye view of cellular respiration. Glucose is going to get chemical energy gleaned off it, chemical energy in terms of ATP and NADH. The NADH actually gets used later to make even more ATP. And overall, the glucose is going to be released as CO2 later on. And now we're going to actually look at the formula and see how it's reflected. We see our glucose here as an input, and we see our CO2 released as a byproduct, and we see a ton of ATP being made. In a bit, we're going to see how the oxygen and H2O play into things once we see the whole process. So before we look at the whole process step by step, we're going to take a quick look at the organelle in which cellular respiration takes place. Also, guys, if you didn't know, organelles are to a cell in the same way that organs are to a human body. They're small little membrane-covered things that have a specific function in the cell. The mitochondria's function inside a bigger cell is to generate ATP. And the mitochondria is where cellular respiration occurs. So the parts of the mitochondria are the outer membrane. And guys, if you didn't know, a membrane is a layer that lets some substances through and helps stop other substances from coming through. So it's selectively permeable. The outer membrane is right here. The inner membrane has many folds, and these folds are called cristae. And we'll see why it has so many folds later. It's to help make this membrane as long as possible for the limited space that it's given. And the intermembrane space is that space between the membranes. It kind of makes sense from the name intermembrane. And then in the very inside, we have the mitochondrial matrix. So it's helpful to gain a sense of where you are in the mitochondria during this process if you memorize these parts. So now we're going to look at the steps to cellular respiration, which are going to be glycolysis, Krebs cycle, and oxidative phosphorylation. And we're going to look at where they occur as well. The glycolysis occurs in the cytosol, or that outer liquidy part of the cell. Because there's a bigger cell around here, there's just liquid around these organelles. That is called the cytosol, and that's where glycolysis occurs. Then all the components enter the mitochondria, and in the mitochondrial matrix is where the Krebs cycle occurs. And then lastly, oxidative phosphorylation is going to occur right along the membrane. So let's take a look at our very first step that's occurring in the cytosol, and it's called glycolysis. Glyco is a prefix that usually refers to glucose, and lysis, or to lyse, means splitting. And as the name suggests, glucose overall is going to be split into two molecules known as pyruvate. At the very beginning, there's an energy investment stage. ATP actually gives energy to glucose, and as you can see, the ATP gets turned to ADP, and it's releasing energy into this glucose. But that's okay because, by the way, guys, this is a simplified version with just what you need to know for AP and college bio. The glucose being rearranged and changed throughout this line right here. There's an energy payoff stage where a total of 4 ATP are generated, 2 NADH are generated, and 2H2O are released as a byproduct. Our net results after the glucose splitting is that we get 2 ATP because 2 ATP were lost and 4 ATP were gained. We get 2 NADH and we have 2H2O as a byproduct. Up next, we're going to be heading into the mitochondria, into this mitochondrial matrix right here for our Krebs cycle. So the Krebs cycle is also a really complicated cycle with a bunch of intermediates and chemical reactions going on. But you don't have to know every single step. What you do need to know are the general, really important general concepts. Those general concepts being, number one, pyruvate oxidation. Oxidation, if we remember from before, that means pyruvate's going to get electrons taken off of it. Sometimes pyruvate oxidation is referred to as its own step, separate from the Krebs cycle, and sometimes it's referred to as part of the Krebs cycle. Pyruvate's going to get oxidized or get electrons taken from it, and it joins with and has 2 carbon dioxide released from it. It joins with coenzyme A to create acetyl-CoA. This molecule right here goes through the Krebs cycle, meaning it gets CO2 taken off of it and a lot of chemical energy gleaned off of it, a little bit of ATP and a lot of electrons. And then what you're left with right around here joins with the next 2 pyruvate and the coenzyme A to create acetyl-CoA again and go through the cycle all over again. And that's why it's a cycle. And one thing that I found helpful to memorize was 4262. And for some reason that helps me remember 4 CO2, 2 ATP, 6 NADH, and 2 FADH2. Oh, also this is our first time seeing FADH2. And what it is a molecule that works a lot like NADH, and that is, it is an electron holder. And we're going to see NADH and FADH2 play similar roles as electron holders when they all release their electrons later on. So basically, we've gotten rid of all our carbon. We've gotten all the ATP that we could from just directly from the glucose. So now what we have is a ton of electrons. And now we're going to see how those ton of electrons are going to be used. So we got 6 NADH from the Krebs cycle and 2 FADH2, right? And actually, earlier, what this should be is actually 10 because we have 2 from glycolysis and 2 from the Krebs cycle as well. We just have a bunch of NADH and FADH2. Remember that concept earlier about how if a molecule with less pull of electrons gives away its electron, the overall system releases just a bit of energy? That's actually at play in what is called an electron transport chain. What this is a chain of proteins, each with more pullover electrons than the last. And these proteins are embedded in the inner membrane. So when NADH and FADH2 drop off all of their electrons, the electrons are actually going to fall from protein to protein as each protein has more and more pull over electrons. And what this means is that energy is going to be released with each of these falls. So what are we going to do with all this energy? What's the purpose of each of these mini energy releases? Well, remember all those protons from earlier, where each time dehydrogenase is at play to create NADH, a lonely proton gets released? The Krebs cycle, which released the most NADH so far, was occurring in the mitochondrial matrix, and that means we have a lot of protons floating around in our mitochondrial matrix. And what we're going to do with that energy is we're going to actually push all of the protons out into intermembrane space. Now, why would this require energy? I want you to remember that basically everything inside a cell, every space like this is really just liquidy substance where particles can float around. Protons like this in a watery liquidy substance just want to diffuse. Naturally, the protons want to be about even on either side of the membrane. But what we're going to use our energy to do is force them all onto one side. And because the protons don't want to do this, it requires energy to make this process happen. So now what we have is a bunch of protons floating around our intermembrane space, and they really, really want to rush back in and even out how many protons are on either side. And that means that there's a lot of potential energy that could be harnessed right now. Because if these protons were given a chance, they would rush back in, releasing a lot of energy. So what we're going to do is use ATP synthase. ATP synthase is another protein embedded in the membrane that harnesses the power of the protons coming back through to generate ATP. It's using that to do another process that requires energy, which is putting that phosphate back onto the ADP. It works a lot like a turbine for a river of protons. And what ATP synthase does is that it generates 32 to 34 ATP per glucose. And this is by far the most ATP generating step. And by the way, guys, this uneven amount of protons in the intermembrane space compared to the mitochondrial matrix that we created is called a proton gradient. And the use of all these protons out here rushing back in as a source of energy is called chemiosmosis. Those are two important terms to know also. So now we've gotten all our energy, we've used up all our glucose. We just need to lastly learn how all these byproducts that we have right now, like all those extra protons, all those extra electrons, get turned into something that can just be released. So what happens is we've seen how carbon here ends up in carbon dioxide. We've seen the how the ATP to get generated. We're going to see how the oxygen and the water play a role. So at the end, when all these electrons are leaving, what actually happens is that oxygen comes in and it splits up. It turns from O2 to just individual oxygen molecules. It accepts these extra electrons and it accepts these hydrogen ions or protons. What happens is H2O means two hydrogen atoms and one oxygen atom. So that one oxygen atom joins the two hydrogen ions and the two electrons to create H2O, and that's what you see in the oxygen being consumed and the H2O being released. And an important phrase to learn is that because the electrons get passed down this electron transport chain and then finally end up with oxygen, oxygen is the final electron acceptor. So here's our sort of overall. I remember my teacher saying it was really important to memorize the inputs and outputs, and I agree with that so far in my experience. But I would also say that you don't really need to know the numbers, but that is just my experience. Take that at your own risk. I always found it really helpful to know the names of what goes in and what goes out. For example, what you really need to know about glycolysis is that one glucose enters and two pyruvate leave, and you get ATP and ADH and H2O. Now, one really frequent question that I saw, like on every quiz and every test, is that the electron transport chain, they would always ask you if the energy coming off of it is used to generate ATP, and the correct answer is actually no. Because if we see from here, energy that we had right here coming off of the electron transport chain, we don't see any ATP synthase receiving this energy. This is not the energy that gets used. What the electron transport chain does is create a proton gradient, which then transfers its energy over to the generating of ATP. So the correct answer is actually the electron transport chain does not generate ATP, and it is the proton gradient that generates ATP. So far, we looked at aerobic cellular respiration. This means that oxygen is part of the process where we could see that oxygen was the final electron acceptor. But there are other forms of cellular respiration that you could be tested on, just a lot less, because aerobic cellular respiration is the main focus. It's still helpful to know these other forms of cellular respiration. And it's basically all of what occurs when oxygen is not present. So when oxygen isn't present, we have a couple of options. Number one, there are some adapted bacteria for anaerobic or oxygenless environments, which undergo anaerobic respiration, which is when sulfates or nitrates, which are molecules heavy in oxygen but just not pure oxygen, replace the role of oxygen as the final electron acceptor. And then we have fermentation, which occurs in other bacteria, other microorganisms, and humans. It's basically glycolysis and then wrap it up. And there are two types of fermentation that you're going to want to know for those courses, which is lactic acid fermentation and alcohol or ethanol fermentation. Ethanol is a type of alcohol, and alcohol is a chemical group with hydroxide. So, as you might imagine, the fermentation, which is just basically glycolysis and a wrap-up, it's going to be a lot less productive than cellular respiration with an oxygen involved, because we only see 2 ATP. So it's going to be way, way less efficient. As you can see, it is pretty much glycolysis and then a wrap up. Except no water just gets released, just those little 2 ATP and that NADH, and then it all gets put right back into the glucose. It varies based on how you create the byproduct. If you take out the 2 CO2, you get ethanol, and if you don't, you get lactic acid. Lactic acid is actually occurs in humans. If you're exercising, this means your muscles are creating ATP to help you move around. And if you're exercising, you're moving around a lot, and you're not getting enough oxygen to those muscles in time. And then you're going to start feeling what's called the burn or lactic acid buildup, because your cells are undergoing fermentation because they're just not receiving that oxygen in time to generate all that ATP necessary. And that should be pretty much everything you need to know for cellular respiration. I wish you good luck on your test or quiz or anything else that you have coming up. Have a great day.